Yes, dear students, Assalamu alaikum. Assalamu alaikum, dear students. Today we are going to study the basic structure of the cell, and first of all, a basic concept that the cell is the structural as well as functional unit for all living organisms. All the structures of multicellular organisms or the bodies of multicellular organisms are composed of cells, and cell performs the basic function for each and every type of function for living organism. Next, the historical background we have already studied from the assignment. And next, two types of cells. You know that there are two basic types of cells: eukaryotic cells and prokaryotic cells. And as the literal meaning of the eukaryotic cell indicates the presence of nucleus, U means true. Karyotic is derived from karyon, meaning nucleus, having true nucleus. So cells having true nucleus is the literal meaning, but actually the basic concept is that the difference between prokaryotic and eukaryotic cell does not lie in the just lies in the presence or absence of the nucleus, but it lies with respect to the all membrane bounded organelles. That cells having all membrane bounded organelles are called eukaryotic cells. And membrane bounded organelles include nucleus, mitochondria, chloroplast, endoplasmic reticulum, etc. All these are present in eukaryotic cell. And number two is the prokaryotic cell. Prokaryotic cell lack all membrane bounded organelles including nucleus. So all membrane bounded organelles will be absent in prokaryotic cell including nucleus. So this will be a single type of cell and it will contain only non-membrane bounded organelles. Next, we will move towards the variety of organelles. You can observe here that organelles are divided into two main categories: membrane bounded and non-membrane bounded. Membrane bounded organelles include nucleus, which controls the whole activities of the cell, so it is known as control station of the cell. Next, endoplasmic reticulum, which is further of two types: smooth endoplasmic reticulum, which is involved in protein synthesis, and rough endoplasmic reticulum, which is involved in the lipid metabolism. Next is the Golgi complex. Golgi complex is also known as Golgi bodies or dictyosome in case of plants. We will study this in detail in the next lectures of the Golgi complex, and it is concerned with the cell secretions. Out of the cell, the materials which are secreted out are basically secreted with the help of Golgi complex and endoplasmic reticulum. And next, plasma membrane or cell membrane or plasma lemma, which is the outermost boundary of the animal cells only, and all other cells have cell wall. So this is a basic concept you have to remember in a short way, that only animal cells are exceptional case in which the outermost boundary is plasma membrane. Other than these animal cells, plant cells, cells of algae, fungi, etc., bacteria, these will have cell wall as a outermost boundary, but the animal cell will have outermost boundary in the form of plasma membrane. Next, outermost boundary of the protoplast. Protoplast is the living matter of the cell. So plasma membrane is the outermost boundary of the protoplast, and all the layers which are present outside the plasma membrane are not included in the living content or protoplast, like cell wall, slime capsule. All these are the substances which are not included in the protoplast. And next is the mitochondria, which is the site for aerobic respiration, and it also performs the process of phosphorylation. Always remember कि केमिस्ट्री में हम जो वर्ड यूज़ करते हैं फास्फोराइलेशन, वो किसी भी चीज़ में फास्फेट के एडिशन के लिए इस्तेमाल कर लेते हैं या फास्फोरस के एडिशन के लिए। बायोलॉजी में जब भी वर्ड आएगा किसी जगह पे, तो यहाँ पे आपने बात ध्यान में रखनी है कि इससे मुराद जो है एटीपी एडिनोसिन डाइफॉस्फेट में फास्फेट के एडिशन है, जिससे एटीपी मॉलिक्यूल सिंथेसाइज होंगे। सो फास्फोराइलेशन in biological sense means synthesis of ATP. Synthesis of ATP is the main function of mitochondria. And next, chloroplast, which is the site of photosynthesis, it is involved in the production of food. Next, second category will be of non-membrane bounded organelles, which are without membrane, and these are number one ribosomes, which is the factory of protein synthesis. All these organelles will be discussed in detail in the next lectures, inshallah. We are just taking an overview at this stage. And next is the centrosomes, which is the clear zone around the centrioles. So the centrosomes or centrioles are non-membrane bounded structures which are present in the animal cell, and centrosome is the collective term for centrioles also. we use centrosome in different senses. It may be considered as a zone around centrioles, or we consider it as a collection of centrioles. Next, it plays a role in the positioning of the spindle fibers, not synthesis. Synthesis of spindle fiber, always remember, does not occur with the help of centrioles. Just they orient the spindle fibers so that the spindle fibers are always produced in between the pairs of centrioles, two pairs of centrioles. And next, it also determines the plane of cell division और क्लीवेज फरो. जिस तरफ दो सेंट्रियोल्स होंगे हमेशा उसके दरमियान से सेल्स जो है वो हमेशा डिवाइड करते हैं, लिहाजा सेल ने किस प्लेन में डिवीजन करनी है, क्लीवेज फरो किधर से डेवलप होनी, इसका तयन कहाँ से होता है, सेंट्रोसोम या सेंट्रियोल्स से. नेक्स्ट, फ्लैजेलम. फ्लैजेलम हैज़ द फंक्शन ऑफ लोकोमोशन and other function is the process of chemotaxis, movement of bacterial cells mainly or any other cell in response to the stimulus of chemicals is known as chemotaxis. Next is the cell wall. Cell wall is absent in mycoplasmas, a group of bacteria, and also it is absent in animal cells. These are non-membrane bonded organelles. And next, distinct organelles of prokaryotic cells. Those organelles which are present only in prokaryotic cells, and these are absent in eukaryotic cells. Number one, plasmids. Plasmids are small circular double-stranded DNA molecule. Actually, these are extra chromosomal circular rings of DNA which are present in some bacteria. It means that these are not present in all bacteria. Number two, these are the structures or organelles. These are the chromosomal rings, DNA rings or chromatin rings, which have additional roles in the bacterial cells. If these are present in all bacteria, it means that these have some important genes which are very necessary for the metabolism or other important features of the bacterial cell. But as we know that these are present in specific bacteria only, so these rings of DNA contain the genes for the special properties of bacteria. And these are relatively smaller, as it is indicated here that the total base pairs in the plasmid may be just 1000 or 30, ranging from 1000 to 30,000. These are much smaller than the main chromosome of bacterial cells. And next is their function. As we have discussed that they have special roles and they provide special properties to the bacterial cells. Number one, antibiotic resistance, and number two, antitoxin protein production is their role. And they also act as vector in genetic engineering technology, in which we transfer genes in bacterial cells with the help of plasmids. And next function is the process of conjugation. During the conjugation process, they pull the main chromosomal DNA from donor bacterium to the recipient bacterium. So these are the organelles of the prokaryotic cell or bacterial cell which are present only in the bacteria. You will remember them with reference to the objective point of view. And next important points for objective: largest eukaryotic cell or ostrich egg. And in this point, you have to remember one thing, that ostrich egg is no doubt largest eukaryotic cell, but when it is in a unicellular condition, only the cell of zygote is present in the egg. At that time, we can say that it is eukaryotic cell, largest eukaryotic cell. But when it becomes a multicellular mass, or it becomes an embryo, at that stage, no doubt the egg will contain many cells, and this will not be considered at that stage the largest eukaryotic cell. So ostrich egg is no doubt and no doubt largest eukaryotic cell, but up to unicellular stage only. And next, longest eukaryotic cells are neurons or nerve cells, and smallest eukaryotic cells are sporozoites and sporozoite merozoites. These are the stages of the malarial parasite Plasmodium. And next, one important thing also, that always the prokaryotic cells are smaller in size as compared to the eukaryotic cells. Eukaryotic cells are larger in size and prokaryotic cells are smaller in size. And next, non-membrane bonded structures absent in prokaryotic cells. As we have discussed earlier, that prokaryotic cells are those cells in which all membrane bounded organelles are absent, but they have non-membrane bounded structures. But this is exceptional case, that some organelles which are non-membrane bounded, but still they are absent in the prokaryotic cell. and these are centrosomes or centrioles. These are absent in bacteria, and microtubules or cytoskeleton. So remember these points, that instead of being non-membrane bounded structure, these are absent in prokaryotic cells. Okay. Next important thing, how prokaryotic cells perform function in the absence of membrane bounded organelles. Like that no chloroplast is present in bacterial cell or cyanobacterial cell, but they still perform, many of them still perform photosynthesis, and mitochondria is absent in prokaryotic cell, but we observe many aerobic bacteria which are present in our environment, how they perform the process of aerobic respiration, photosynthesis, cell secretion, etc. This is an important point, that no doubt they lack all membrane bounded organelles, but the invaginations of the plasma membrane, which are known as mesosomes, these have enzymes for these activities like respiration, photosynthesis, cell secretion. So actually the invagination of plasma membrane mesosomes perform all these activities or functions of the all membrane bounded organelles like mitochondria, endoplasmic reticulum, Golgi complex, and chloroplast, etc. Okay, you have to remember this point. Okay, that's and from the variety of organelle. Next, we will move towards the next point. Okay.